Let's talk about the goals of DNA sequencing methods. Learning objectives. This chapter will demonstrate how to understand the goals in data collection for DNA sequence collection and variant identification. Compare and contrast the following methods: DNA-seq microarrays, whole genome sequencing, whole exome sequencing, and targeted sequencing. Recall that mutations are just changes in the DNA. Sometimes these are single base changes listed in this picture, but sometimes DNA variants are long sequences of DNA that have been inserted again and again, like copy number variants. Living cells add mutations all the time, and oftentimes these mutations are caught and repaired, but sometimes they continue on. Mutations that carry on might be harmful or pathological depending on where they happen in the DNA and what types of genomic features they affect. Remember, DNA leads to RNA, which leads to protein. So in the situation where a DNA sequence affects the output RNA or DNA, this might change the functionality of that RNA or protein, which, in turn, may affect the functioning of the cell. Most often mutations are harmless because the overwhelming percentage of the DNA is repetitive sequences that, to our current knowledge, do not have strum functions in the cell. In other words, the percentage of DNA that leads to RNA or protein products is very small, around 1–5%. This doesn't mean the rest of the DNA doesn't have function, just that its function is less clear or less direct in its effects on the cell. The goal of DNA sequence analyses then is usually to identify DNA variants on a genome-wide level. This is usually done by comparing DNA sequences to a reference genome. Types of sequencing: whole genome sequencing was try to read everything. Whole exome sequencing was try to read protein coding sequences. Targeted sequencing only read things at locations we are aiming for. Comparing was to other sequencing modalities. The most common DNA sequencing techniques are described. Whole genome sequencing coverages all genes and non-coding DNA. 2 billion bases are covered when applied to human samples. This is the most expensive of the techniques. Depth of coverage required for 99.9% sensitivity is X. Whole exome sequencing coverage is the exome or expressed genes. Approximately 45 million bases are sequenced. This is a cost-effective technique. The depth of coverage required for 99.9% sensitivity is X. Targeted gene panel sequencing coverage is 5,500 genes. 0 to 62 million bases are sequenced. This is the most cost-effective technique. Depth of coverage is X. There are 3 general methods we will discuss for evaluating DNA sequences. Whole genome sequencing assays more of the genome than other methods, but is much more costly and computationally intensive. Depending on your goals, this may be overkill. The microarrays, on the other hand, are much more cost-effective, but are not able to be used for exploratory purposes. Whole exome sequencing WES and other targeted sequencing methods allow you to survey regions of the genome in a way that is more cost-effective and potentially at higher depths. DNA sequences can actually be used like a biological signature to identify a person. If you are working with human DNA, not cell lines, this is a protected data type. Please consult your ERB and other data privacy experts when handling your data.